but he actually signed papers as Cajal. Okay? So I think it was S dot R dot, so S dot Y R dot Cajal. Probably he was actually sorting for foreigners, so they had less of a...
For citation purposes.
So my guess is that it's taken after that. Or if not, you have another anecdote.
Okay, go ahead.
Okay. So it's a pleasure to be here. I was actually making the comment before that I've been talking to a few people today, and I've been talking about science more than in my institute for the last 4 years, because the type of things I'm doing are slightly different to the type of things the institute is doing, but there you are. So I started to study things like that, so a neuron, and I'm moving now since I got a permanent position in 2008 to study groups of animals. So the lab still is publishing mostly in other things, because we were working on those a few years ago, but we are thinking about decision making in animal collectives. So let's go for it. Let me just summarize very quickly the type of things we are publishing, so the things we were thinking about a few years ago. We were interested in trying to understand, we would call it theoretical neuroanatomy, why neurons are in the places they are. And one can actually assume that the total length of the nervous system might be minimal, and that seems to be working fairly well, at least for the systems we've been working with, Drosophila, C. elegans. Before that, I tried to understand simple problems in how neurons process information. There, we would be using things like information theory. But as Maxi was telling you, I'm drifting to trying to understand the decisions of animals in groups, and also trying at the same time to understand that at the level of brains, how brains play a role in decisions. Of course, they do. I'm trying to understand how that happens. So the only work I've published that has to do with things I will explain today is this closed computational biology from the end of last year. I'm not going to speak about that because it turns out to be a very particular case of the things I will explain to you today. So today I'm going to explain you things that are so far unpublished, although if I don't screw up in the very last second, it should be published very soon. The type of problems I'm interested in are illustrated here. Like I have one fish. I work with zebrafish, trying to understand, trying to work out whether that's a predator or not. Zebrafish is an animal that actually moves in groups. So not only has this information, but the information of what the behaviors of other animals are. So in this case, if this guy is escaped, well, that increases the probability that that might be a predator. Okay. So that's the idea behind the types of things I will be showing to you during this talk. The basic hypothesis I will be making, to move on, is that the group behavior actually is a result of individuals estimating the world using non-social data and social data. Basically, I'm going to make the assumption that all the things that happen in the brain in a situation like that, in the end, are very similar to a calculation of the probability that there is something out in the world, let's say a predator, given what you see, that is not exactly a predator, but something more complex, more ambiguous, and also the behaviors of other animals. Okay? I'm exaggerating a bit. In Photoshop, I open the mouth of this guy, so it looks like they are escaping. But the idea is that the brain uses the information coming from the behavior of the animals to estimate the things about the world. Right, so I'm not going to go through all the maths in detail. I will not go through the derivations. They are in the papers. They are in archive. But let me tell you what are my basic assumptions and the basic theory I use, okay? So you can already criticize me at that level. The type of problems I'm going to deal with is that I will have my animals, trying to decide between two options, X and Y. And there will be other animals that have already chosen X, other animals that have chosen Y. I'm going to make the assumption that, as I was telling you before, each animal is computing the probability that X is a good option given what they see and given the behaviors of other animals. And the same thing for Y. I'm going to assume that the brain of this guy is calculating the probability that Y is a good option given what they see and given the behaviors of other animals. Okay? Now, this is the step I'm going to skip a bit. It's in the derivation in the paper. So what we're going to do is to express these two probabilities using Bayesian theorem, and rewrite the probabilities as some parameters. And it turns out that the probability you get after one simple approximation, that I do it for the sake of explanation to have some simple formulas, I will show you data without any assumptions, is of this form. So the probability that X is a good option given what you see and given the behaviors of others is of this, has this format, where A is actually the probability that X is good given what you see, so it's non-social information, and the social information comes here. So S is how reliably other animals are telling you that X is a good option, and NX is the number of animals that have chosen X, and NY the number of animals that have chosen Y. Okay? So the same for Y.
Okay.
K is actually, so let me show it to you. K has all this format here. So basically it measures a relation between how well your, so let me see what letters I'm going to use here. This is all wrong actually. So this is choosing X, but actually I think it's, so actually what it should be is, let me see. I think it's here a capital B, so the behaviors, how the behaviors of others relate to Y being a good option or a bad option, relative to how they indicate that the other one is a good option and a bad option. So basically it's a term that when you calculate the probability that X is a good option, how the animals choosing Y might be telling you that it's a bad option. It might be zero, it might be telling you nothing, or it might have an effect that is as high as the animals choosing X. But I'm sorry, the formulas here, probably I did copy-paste, and they all look very much the same to me. Let me go back, because there's a strong assumption I'm going to make. So this is the estimation probabilities, and actually when we started, we assumed, I didn't read anything.
May I challenge the basic hypothesis?
Yeah.
I mean, I'm not sure what you mean by non-social. In the absence of social interactions, you know nothing.
No.
I mean, if you isolate an animal or a person.
But that's not the idea. It's not the idea that in the past you've been isolated and you've been in a little square suffering. The idea is that for the given behavior, what you would think about whether that is a good option and that is a bad option if you don't have information from others. So in an experiment, it's a zebrafish given two options and a single zebrafish. What does it do? What happens is that in the symmetric case, the probability is 1/2. In the asymmetric case, different from 1/2. So if it's a darker region than another one, fish prefer darker regions. So that A will be a preference for the darker region. For the symmetric cases, it's basically 1/2. So when we started to do this, we assumed that when you calculated the probability that, so you calculated these two probabilities, basically we said, well, if the probability that X is a good option is higher than the probability that 1 is a good option, then choose X, otherwise choose Y, okay? So if this is with higher probability a better option, then choose X. And we couldn't feed any data for a few weeks. We didn't know what to do. And Alfonso Pérez Escudero, the PhD student working on this, one day comes and says, Gonzalo, it turns out that if I say that the probability of choosing X is just the estimated probability plus this is normalization, this in the denominator, all the data is fit. Okay? And I said, well, yeah, how are we going to publish that? It turns out that this is known in animal behavior as probability matching. This is still mysterious. There are some theorems that say, well, it turns out that when probabilities are changing in time, this is an optimal solution. It's not clear actually why animals do this. Humans also do it. You don't have lots of time to learn about a task. They do probability matching. Okay. So it's known in animal behavior that for many tasks that have nothing to do with collective motion, but tasks of decisions of a single animal between options, animals use probability matching.
So there's something I don't understand. What is the normalization of the first equation, which then relates to the second?
Actually, this is not normalized because it's actually quite interesting. In the first paper, we were interested in capital X being is the best option. Okay? And then the probability of Y is 1 minus that. But now we change, because that didn't work that well, to X being a good option. So the both options could be good. Okay? This is what we are doing now. Both options can be good. Okay? That's perfectly fine.
The second equation is just a normalization of the first one.
Sure, it is just a normalization. That's what probability matching is. And it's illogical. It's illogical. Yeah, let's understand it. Let's understand it. From a mathematical point of view, it's just normalization. But rational decision making says, with time, this is happening to me. That what's rational is starting not to look that rational. Is that if the probability that X is a good option is higher than the probability of Y being a good option, choose always X. This is an algorithm, and this is a different one. Is the probability of going to X is given by this. So this does not need to be higher than the probability of Y being a good option. It goes there with the estimated probability. That is what is not obvious. The behavior is go to X with a probability that is proportional to the estimation that is a good option. So this is behavior, this is estimation. So it's the simplest assumption if you wish, because it's a normalization, but it's actually completely non-trivial. Is that the behavior goes with estimation. Most people, all the literature says this. There's lots of modeling about copy in social settings that will say this. It's called information cascades.
So what you say is that animals behave like particles. They don't take a decision. I mean, if you have a particle that has a probability being in a state A and a probability of being in a state B, whether the state A is more favorable or not, if there is a probability of being in both, it will be a fraction of the particle in both states. And that's what animals do. If there is two options and there is a probability of choosing any of them, they choose the option in the same probability. It's not
but it's not the probability of choosing. This is not
they don't behave rational. They don't have this thing. They say, no, if this is bigger, all of them will go to that place.
Yeah, I can phrase it like that. I would just make the difference that this is not the probability that X is a good option. It's the probability that, so it's not the probability of doing the behavior, it's the probability in the estimation, the probability that X is a good option.
Yeah, I mean, okay.
Yeah, you can.
I could say that this.
I have two objections. I object something called P, where is not a probability. So the first line, P is not a conditional probability. Otherwise, it would add up to 1. The denominator would be 1 with the working probabilities. So this P is something, okay, which is not a probability. Otherwise, the denominator of the second equation would be equal to 1. No, the denominator of the second equation.
This is a number that goes from 0 to 1.
But it's not a probability.
Why is not a probability?
Because they should add to 1. px plus py should be true.
No, because both options can be good ones.
Are there more options? Are there two
They
options?
can both be good options.
But there are more options or just two?
No, there are no more options.
Then
But
they should not do one. Then the second.
no, wait. Can I answer to the first objection?
It's not that.
I'm saying that this is the probability that X is a good option. So you would be right if X is the best option. Then Y would be 1 minus X. But X is a good option. Can you have in front of you two options that are good? Yes or no? With probability 1, yes or no? There you are. Then there are probabilities, and they are for good options. And here is the behavior that needs, in the end, when you count numbers, in behavior, needs to be normalized. But both options can be good.
We have here two good options, which is to continue the discussion, which is my normal way of doing
Okay.
it, or to let anyone know that you have to catch a plane. So explain what you wish to do and then see what time we have for this.
But what is true is actually that this looks very simple, but conceptually it's not non-trivial. Okay, so yeah.
That also is telling you that the behavior, I mean, if Px is a probability, that means that if you are in front of the same situation twice, then you could do a different thing.
Yes, of course.
No, different.
Well, it's a probability. So in front of the same situation, a probability tells you that 2 times.
And that's true. I mean, if
That's of course it's true. Yeah.
even for humans.
Even for humans. Yeah. Yeah.
Okay, let's choose the good option that you go ahead and explain us what you want to do.
Okay, so let's look at what you obtain. Then you have the function that you get is of this form. Let's plot it to see how it looks like. This is the probability of choosing X, given that nx animals have chosen X, and given that ny animals have chosen Y. And the plot is quite weird. It has a high probability here, which is normal. So given a fixed number of animals at Y, this is just a function that increases with the number at X, which is very normal. But the general plot is of this type that has some particular features. So the first particular feature is that it has very different behaviors at low number of animals than a high number of animals. Okay, different behaviors here. And also that it has a simple slope given by k that can be from 0, for example, to 1. That would be a complete diagonal. Okay, so what the claim would be if one tries to make a theory that is not specific for an animal is that different graphs of this type would be fitting different animals or different experimental settings. So let's check it. So let's start first with an experiment in zebrafish. The experiment looks like this. So you have a single animal choosing between options X and Y, and you have different animals in X and different animals in Y, NX and NY. So it turns out that the one that works well is the one that k equals 0, which is a funny function because if you have you are a fixed Y, it increases with X. But here it's quite strange because what happens is that with a number of animals at Y, let's say 3 or 4, when you increase the number of animals at X, X doesn't win. It goes to a probability 0.5. The logic behind this is that the relation between number of animals and each option to be a good one is a nonlinear function. And what happens is that already 4 animals at Y tells you the same thing that the 8 animals are 8. They are both telling you that both options are fine, probability close to 1, and then when you normalize, it goes to 1/2. Okay, you do the experiment 20 times, or for 20 different animals in this case, and you get this value here for this experimental situation, 3 at X and 1 at Y. I change here the order just for the sake of comparison with data, it's simpler to see. when you do the experiment for many different configurations, which in the end is around 300 animals, this is the data compared with the surface you obtain when you fit to the data, and if you look at particular lines at different NYs, you get this comparison here. So it works fairly well for the data. Right, so it turns out then that at least the experiments in zebrafish we've done are fairly well explained with a function like this for k equals 0. Now, once we did the experiments, we said, okay, maybe we can get other data sets explained by the same theory. So we asked people for data in different animal species, and there were two cases with large data sets, the case of ants and the case of another fish species, sticklebacks. So this is a curiosity slide. So maybe you know about Weber's law. It was first discovered for weights. So what happens to us is that when you have a weight of 0 and a weight of 10 grams, you know that 10 grams is more weight than 0. But when you have 100, 110, you don't look at the difference. You don't see this one higher than this. You almost see the same weight. So it turns out that you are not looking at differences, but relative differences. And actually, if you think about it, relative differences in this plot goes at straight lines of slope k, which is the prediction. So we should be seeing in some animals Weber's law in decision making in animal groups. So we took recent data published in PLOS Computational Biology in 2012, in which you had Argentine ants moving around, and they recorded in video how the ants were choosing between X, left, and right. And they also recorded how many ants you had before at X and Y that is assumed to be proportional to the pheromone they are living there. So you plot then the probability of choosing X in the case of ants, like in the plots before, against NX and NY. This is what you get experimentally. And for the theory, you would get fit to the data like this. The interesting thing, as I was telling you before, is that you would make, well, first they had something like that slope k, but also in the theory, as it is the case in general, the low end behavior and the high end behavior are different. So in order to check for that, one can plot the function in a different way to exaggerate this region here. So it's a different way of plotting this. Nx minus ny divided by the sum. Here the sum, so this slope here goes into a horizontal line, and this thing gets really big. And you look at the data, you also observe something similar. So again, in the data you observe a difference between the low end behavior and high end behavior. If you want to do something slightly more quantitative, what you can do is to divide the experimental plot and the theoretical plot in these diagonal lines and fit the probability along those lines and plot the slope of that fit. And the Weber law implies this black line here. What you see in the data are these blue dots, and the theory here will imply this like here. So it really implies a Weber law for high end and deviation for low end that fits reasonably well with the data. Okay, so as I told you before, actually one thing that doesn't seem to work well, if you want to look at details, is that the theory here is fairly flat. This region, the probability is 1/2, whereas here is a bit more smooth. If we don't make some assumptions that we use in obtaining simple formulas, but use the fact that all animals are using the theory without any approximations, we obtained a bit more gradient here, more similar than in the experiment. But that's
a question not to discuss, but to understand. Yeah. You are telling us something about the value of your parameter k. You said that a is essentially 1/2, but you don't tell us anything about the parameter s.
Yeah, I think here was let me remember.
But you have to fit?
You have to fit this. You have to fit this. So you have, in the end, the 3 parameters here, which 4. But then with those, you explain all the data sets. It's not. So for each data set, you can create formula that have less parameters. Actually, not that easy for the 3 data sets I'm going to show you that you can do with less. Okay, so with this same function, we've been just by looking at different shapes of this function, it seems to be fitting reasonably well experiments in zebrafish and experiments in ants. So let's try to look at another data set we had. It was another fish species, sticklebacks. And the experiments were quite nice. These guys, Ashley Ward and collaborators in 2005, that we used the data in a current biology in 2008, had the following setup. They had a tank in which they had a number of real fish, real sticklebacks, and they produced replica fish made of plastic that were moving in lines to X and to Y. And they were, in the end recording, they were repeating the experiment many times, and they calculated, of those many times, 20, the proportion of fish that were going to X. So what does it mean? A proportion of fish going to X of 1 means that all the real fish go to X. And the proportion of experiments in which that happened was half of the experiments, 10 of them, maybe 11. Then a proportion 0 means all the fish going to Y. And a proportion 0.5 is, in this case, 1 fish going to X and 1 fish going to Y. Okay? So you have these finite probabilities. So in our case, what we can do is apply our model sequentially to see what finite probabilities we get. And in this case, using k equals to 1, but again, using also a value of s, if I remember correctly, is 2.5. You get that the model tells you that for different group sizes and for different replicas, you should get different answers. So for a group size of 2, 4, and 8, and 1 replica going to Y, 1 replica going to X, 2 replicas going to Y, 2 replicas going to X, 0 replicas going to Y, 1 replica going to X, and different proportions of replicas. The experiments are at the bars in blue, and the model for some parameters for k equals 1 gives you that fit. In this case, they were using a predator, a plastic predator, and in that case, that changes the value of A. Now the setup is not symmetric anymore. A single fish does not want to go to X anymore with high probability, but given enough number of replicas, it will, in some experiments, go to the so which one is it? The fish going to Y. So half of the experiments basically goes to where the predator is. So just, again, the fit is reasonable, it's slightly worse than in the previous case.
So what is the value of A here?
The value of A, I don't remember.
And now the 2.
What's that?
There is no symmetry anymore.
No, it's asymmetric. Now it has a value to make a single fish go to, so basically you would be fitting, you choose A to, so this can be described with no replicas and a single fish. That tells you the value of A. And they also did experiments in which they had changed the shapes of different of the different replicas to the right and to the left. And in our case, that allows us to change how reliably these fish see the behaviors to compute whether X and Y are good options. And here it's less interesting in this case because there are more parameters you can change, but it still works very well.
So just one question. In this experiment, you say that this number of fish following the most active replica. So there is somehow. Oh, that's what the fish.
Oh, in this one, yes.
So there is a fish that acts as, say, a leader, or that signals something, or that triggers the behavior of the other, in this case?
Yeah, so when you change, but basically this is a way of plotting because you can order it following, so this is a way of saying left or right.
Okay,
and
so
ordering all of them in the same way. It's not more than that. All that happens is that when you change the shape of these replica fish, they are less interesting or more interesting for the real animals. And the reason to write this down is a way of having all increasing to the right.
But that doesn't mean that there is a fish that triggers the other ones to behave, or whatever, the replica fish that takes a decision before the others.
So according to the experiments and according to the theory, what happens is that these more reliably push these guys to go to X or Y depending on the shape. The color, usually what happens is that you deform the animal farther from the real shape, color, or size. They are indicating to the real animals less that the options are good or bad, and they are not follow with the same probabilities. In a model, it changes the value of X. We allow the animals to have, these two animals to have different values of S. Actually, in the theory it works out that it's only the ratio of the values of S that is important. This is a little detail. So what happens in the end is that we derived a function that could have, I would have guessed it shouldn't have worked, because it's not specifically derived for a particular experiment. We first checked it in sticklebacks, and it was working fine. We did experiments in zebrafish, and it was working fine. And we asked for data in ants, and it worked fine. But I'm sure at some point it will break down. Okay, so the second part of my talk is going to be, well, Gonzalo doing experiments in very simple settings in which he is having their animals choosing between two options. Of course, we need to move forward into considering more complex situations. For example, the question of whether there are some animals that are more leaders than others. The question of how you choose in real space and real time. For that, you need to do more complicated behaviors. The problem is that when we started to think about it, and still the situation is like that now, there is no software that automatically gives you the movement of animals in your tank unless you mark the animals, or unless you have a PhD student spending months and months correcting for that. And that's not the way to move forward. I mean, you can publish a few papers, but it's very difficult to make progress in this situation. So we spent 3 years now trying to develop a system that actually solves the problem. And it has been so hard that I'm going to tell you about it, even if it's a bit too technical. You might have extra ideas so you can tell me how to do this better. But so far, I think it's the only software that can do it. So the problem is that when you record from above and these two animals cross, you cannot distinguish that situation in which two animals cross from a situation in which these two animals just touch and separate. So actually, there is no algorithm known that maybe is known to the US Army. It's not published. And the situation in zebrafish is really bad because it's not only simple crossings, but here you have these animals and you see lots of crossings there. It's really complicated. So you can have a student and do it. But let me tell you the history of this behavior. That's interesting. So this is a learning experiment or a conflict from learning. Let me tell you about it. So this is a curiosity, but so I wanted to have a complicated video to show that the tracking system worked. And what we did was we were doing learning experiments, and here you have 8 animals. But what we did during that week was to train 4 of the animals to choose this corner with food, okay? And another 4 animals to choose that corner there, okay? And then we said, okay, let's put them into a conflict. We have the 8 together, what should they do? Should they split? And you saw already what they did. They first go to that square, all of them, and then they go to the second square. So that's how they solve it. Right. So we were thinking how to do this, and it was a bit crazy. So you are a computer scientist, there are many things you should try, Kalman filters, I mean lots of things you can try. We didn't. And probably was good because what we did was to take pictures of the animals, and you can see that Lucy, Tom, and Jerry look different. Maybe you can see that this guy has this different to these guys. So these are slightly higher resolution than what we use in real videos, but you can see that individual animals are different. And we exploit that. So the idea is that animals are marked. We don't need to mark them. Okay? And we just need to recognize the marks. And we need to do it in a way that is robust so it works. Okay, I'll go quickly, but for you to see that from now on, one can obtain lots of behavioral data because it's going to be extremely easy to do so. So we divide the video into what we call segments, which is the frames between two crossings. So the animals, you have a crossing here, a crossing there, a crossing there. This is each of these two things, we call it a segment. Basically, this is not the most complete thing to do, but we take from the whole video the longest segments, and we say that this segment here is fish 1. So these are non-problematic segments because the non-problematic pieces of video because animals are not crossing here. So we call this guy a name or a number, fish 1, fish 2, and all of them. And what we do is we don't think of fish 1 as an average image, but we take the list of frames, all the cards, if you want, of the stack, and we call fish 1 all this stack. Maybe here you have 200 frames, and the same for each fish. Okay, and the nice thing about that is that they have many different postures. So fish 1.
But each frame has many fishes, not only one.
No. Each of these frames is just a cat.
So you cut the movie only on that.
I segment basically the fish. Yeah. But now, to compare two images is a nightmare, and I would not recommend to do it. It will not work. We did it. It didn't work. But you want something, a way of plotting what an animal is that is translational and rotational invariant. So for that, what you can do is something very simple, is to take every 2 pixels on the image of a fish and work only with the intensities and the distance between those 2 pixels. And you can plot something very simple. It's not the most general thing, it's the one that works and works fast. You can work with the statistics of the sum of these 2 intensities against the distance between the 2 pixels. So for example here, fish number 1 will have in this frame these statistics of intensities and distances between every pair of pixels. So before we had a stack of frames for each fish. So we do for each of these frames this transformation. So fish 1 is all these statistics for 1 for each of the frames in the longest segment in the video. And once we have a problem image, we don't know who this fish is. All we do is we do the transformation for that image and we compare with all these frames. And we see that we just abstract them and give a difference. And this guy is closest to this guy. We say, okay, this problem image is fish 7.
Sorry, but this means that the different fishes have different shapes or they have different sizes because, I mean, if all were of the same shape and size, basically there's no difference. This diagram would be the same for all the fishes.
So they have different patterns. That was the idea of the picture before.
But it's just black and white.
What's that?
I mean,
This is grace. This is grace.
No, but you fit the transform in black and white.
What's that? Black and white here?
Yes.
No,
and now you calculate from
it's
this.
grace. It's grace. It's a level.
Okay.
It's grace. No, it's not black and white.
Okay.
So we choose. So we take probably each time we do an experiment, we have 300 fish, and we choose the 8 we find closest in size. Okay. And also we do as a test. We put all of them in black. We do this and it doesn't work. It really needs the patterns.
Probably would be better with color pictures.
What's that?
Would be better with color picture.
Yeah, more time to analyze. Yeah. Seems to work in way. Okay, so for each segment in the video, we go through this same process and we say, okay, blue, 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 or whatever that color is, purple, green, 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 green. Sometimes you get it wrong and gets it wrong, and you calculate the probability that that guy is blue, and you put all of them to blue, and you calculate the trajectory. It's fine. Then the video will look like this. We will do a validation just in a second. Then the video will look something like that. So it doesn't matter what they do, they get recognized all the time and put their colors. Right. So then what you can do is now, as a check, to have a student that goes through the video and compares the algorithm to what he would do, Alfonso again. And let's first try to see what a standard algorithm would do. Well, it will work for a couple of seconds, but already the first crossing it sees, it makes it wrong, and it goes into black if it doesn't coincide with what Alfonso thinks is. And basically, sometimes at random gets it right. But you will need to correct crossing by crossing and put the right colors. Whereas in our case, because we get the animals recognized every time, the errors don't propagate. You can have actually small errors. The first time where they go all here, they resolve it without any problem, but it has some short trajectory that gets it wrong. But it doesn't matter because actually what the algorithm does typically is to say that the probability of assignment is very close to 1/8. It's not very clear which fish is. But anyway, even if it makes it wrong, it self-corrects because it checks again. This is a short. What happens basically is that you have a crossing, very few frames, and another crossing, so you can have a mistake, and it's not. It can happen that you have a mistake in the 3 frames, but with very low probability. So what happens is that 99% of the frames are correctly assigned, and the remaining 1% are very short trajectories.
Classification in real time?
What's that?
Do you analyze them in real time, the video images, or post-processing?
No. This is post-processing. We are now working on that, trying to have as much time on the graphics card and try to do it on that. So far, it's so you see, this could not be done before. So first, we have to do it the hard way. It's a hard computationally. It's hard because you need to calculate all that statistics. But we are trying to get as close as we can to real time. Okay, so. One nice thing about the algorithm is that it can be used, for example, in situations that are much more complex, like you can cover the animals, and in tracking systems that really do follow the animals, if you cover them, you lose completely the trajectory. You can do things like that. You can have complex situations in which the animals get covered, they go away, it doesn't matter, they get recognized from the patterns, that's not a problem. And the nice thing also is that we've done experiments in which, this is the experiments I'm going to show you next, in which you want to work out, for example, who are leaders and who are followers. And the nice thing is that you can do the experiment once and work out who is leading and who is following. But you are more interested in animals that are strong leaders and strong followers. That is, animals that 1 week later they will be leaders again. And the nice thing about this tracking is that it's not only tracking, it's recognizing the animals. So 1 week later we can recognize who John is and who Tracy is and see whether they are true leaders or true followers. So I'm going to show you some very simple analysis just for the sake of showing the type of things you can do with the tracking system. So what we can do, for example, is to work out who is copying the velocity vector of whom. For example, in this case, you have this animal with this unit velocity vector, and this hand guy here takes a while and then to copy to have the same velocity vector. So you can work out with the correlation function with a delay that goes to correlation 1 at a given delay, and you can then work out who is copying the velocity vector of whom. And I can show you in a video how that would look like in real time. This is another fish. This is Medaka fish, a Japanese fish. So I plot in blue if this guy is following this one, and in red if this one is following the other one, at least given by the correlation. But you see that most of the fish are copying this guy here. But you do it an average in time, and you find the following graphs. Let me see if I get this correctly. So we do the experiment in day 1, and the graph means that 2 is copying 5, 5 is being copied by 2, and we do that with all the pairs. So 5 would be the leader according to the definition of the rest copying its velocity vector. 3 and 10 are also reasonable leaders. 1, 2, and 4 are followers. We do the experiment a week later, and it turns out that 5 is not a leader anymore, but 3 and 10 are still the strong leaders, and 1, 2, and 4, 1 and 4 mostly, are followers. And 1 day later, actually, even 1 and 4 get decoupled from the group, and 3 and 10 are still followers. Sorry, leaders. You can also work out who is in front of whom, also in pairs, and plot the distance between the average distance between 2 animals, and you can see actually that there is a tendency for leaders to be at the front and followers at the back of the group, and you can actually plot this out. You can have the relative delay, so these high values are the leaders and low values are followers, and the probability of being in front, and there is a strong correlation between being a leader and being at the front of the group for the DACA fish. Okay? It might be different for the single fish. Okay, so I'm going to conclude. Okay, I think my opinion at least is that we are in a position of us and other groups of starting not only to produce models for decisions in animal groups that fit data sets. You can create always functions that fit, but we can start to produce hypotheses that give you theories that you can test in data sets, and with interesting outcome that the same kind of theory can be quite good at explaining animals that before people had tried to explain their behaviors in terms of very different functions. In this case, just coming from Bayesian estimation, it turns out that the same kind of functions with different values of the parameters can fit very well the data in those cases. I still think that these experiments are very interesting, particularly between finite number. Actually, another interesting thing is that, well, there are several interesting things about doing things in this way. One is that if now someone asks me, Gonzalo, what do you think is going to happen in a setup with four options? I just go to the same formula and I give you it, okay? Whereas if you ask a guy asking for fits, say, well, I don't know how to generalize, there are many ways of doing it. But if you go through the Bayesian estimation and they ask me about 4 options, I say, this is what the theory tells about 4 options. And it turns out that there's a guy in Oxford that actually, in the plus computation biology, we say, we predict that for n options is this formula. The guy actually used it, and it seems to be working for 4 options, which is quite interesting. And Ian Cousin in Princeton is using the same formula for 3 options, it's also working for him. So it tells you for any setup how the formula should look like. And also another interesting thing is that parameters now are not just fitting parameters, so A means something, S means something, there are some probabilities you can play with. So there are experimental manipulations that affect directly the values of those parameters, and then you can say, okay, if I do this manipulation, then it affects these parameters, then I predict what should be the outcome. They are not just fitting parameters. They are, in fact, things that you can manipulate in experiments. Okay, so A has a probabilities inside, S has some probabilities. We can try to manipulate those in experiments. But at the same time, we need to move forward and try to modify this type of theories that actually basically they have their data in a format that it doesn't matter if you have leaders and followers, because the way you do statistics, but in dynamics that's going to be important. It's not only leaders and followers, there are many more things. How do you do decisions in space and time is not trivial. And we did create this way of acquiring data in an easy way to move into that direction, and we plan to publish the tracking system and to make it public so people now can acquire data more easily. And something I'm going to tell you about is that the reason why I started to do these type of things and the reason why we do this type of modeling is that it turns out to be fairly natural within neurobiology. And some of the things we are doing in the lab is to try to work out which regions in the brain are important in social settings. So we have some idea of which brain regions activate when the animal is surrounded by other animals. And the plan now is there are some techniques in neurobiology in which you can generate genetically engineered neurons to express some channels that with light will make the neurons to be active or either to inhibit them. And the idea then is to have only the neurons in particular brain regions to have those receptors and actually to be able to activate or inhibit particular brain regions of some animals in the group to be able to manipulate the dynamics of the group. So if you want to think of an ideal experiment, we would be able in the future to say, okay, we want John to be a leader, so let's shine light on this region and let's try to see whether we can shift a follower into a leader, for example, or to make a fish to see something in the middle of their movement. Okay? We can shine light on particular neurons that have to do with object recognition and make the animal to think that there is an object somewhere. So this is the type of things we are doing on these projects. Thank you very much.
Thank you, Gonzalo. I think we do have a few minutes for discussion, so if there are questions, let's try to make them short and the answers short.
Very populous question. Is there any data or analysis of data related to financial markets, stock trading? So would the same simple rules and probability matching apply to those as well?
Interesting you ask, because so we have one prediction from the field, which is it's actually not intuitive, but our prediction is that when all options are bad, so if you estimate all the options to be bad, according to the model, you should aggregate. Okay, and it's actually not trivial. And we want to look at data of that type to see whether when there is a correlation between the estimation that alternatives are all bad and people doing the same things. We want to test that. We don't have. Yeah.
You have estimated probabilities of an option be taken after observation of the ones that have taken the decision previously. Those that took the decision previously probably have also arrived to it by observing. But how is the process of decision building? It's always start with a leader and then there is some cascade, or have you studied how the decision is, the opinion is constructed and then propagated?
Yeah, so I think we're doing it wrong in the sense that we just allow our animals in the model to do sequential decisions. Okay, so we start one animal deciding, so at the beginning there would be no other animals choosing between X and Y and needs to decide with the algorithm. But once that have decided, we pick another one. So in our case, there is no time in between, it's just discrete decisions, and we only allow animals to do it in a sequence. So we have checked actually that if we allow animals to do it simultaneously, the results don't change to be able to be on the safe side. But we have not done it properly in the building review. That's something that one needs to do. It would be, I've observed weird things in my fish. Like, these are very naive observations, but actually they seem to be doing sometimes like they are quiet and they start to go into some evaporation state that actually makes them a high probability of choosing. So there are weird things in the data that we should put in how this is built, but we are not doing it there at the moment.
Yes.
So you can manipulate the parameters.
And in principle, you could manipulate the parameters.
Yeah, but okay,
So A is simple. A, the parameter A.
So you have some calculations of the parameters, and for instance, why there are differences between parameters for different species?
Why? That's a good question.
But do you have some interpretations?
I wrote something in the paper.
My question, I think, is related. I'm not sure if it is a question or a comment. You have a formula with 3 parameters that with these 3 parameters you are able to feed 3 data sets. That's very nice. Now, in terms of theory, at least from the explanation I've heard today, I haven't learned much about which are the mechanisms that are included in that formulation. I mean, you wrote down a formula at the very beginning that I haven't understood what are the mechanisms that are included there.
Yeah, so I don't know, different people feel that learn different things, but what I've learned is the following. It's actually not terribly obvious to me that the detailed decisions and detailed behavior of a group actually is what I'm saying. So what I'm saying is, for me, not trivial. You are using the behaviors of other animals to better estimate whether different regions of the space are good or bad. Okay? There are many ways, there are many reasons to be in a group. You want to have sex, you want to eat other animals, you can have many reasons to be. You can
Okay, but those are different mechanisms.
Yeah, so I'm putting my money on animals using other animals for the estimation of whether different options are good or bad.
This is herding.
Okay.
So does your formula only account for herding behavior, or?
No, it's extracting information. Is the change in the...
What is herding behavior? Copying behavior?
Follow the ground.
Well, but these are the functions. Do you fit with any herding model the data? My answer is no. So it's estimation.
That I appreciate.
No, but that's the difference. Now you prove it. So you can say, I have a model for why we all dress in similar ways. This is information cascades. And it doesn't fit data. Okay, it doesn't fit data. You may want to believe it or not. It's a matter of
No, but my question, Gonzalo, is if you can go to the first, your very first transparency.
My name?
The one in which you wrote the formula.
Okay.
But I didn't understand, I don't mean that you didn't say it. First formula.
This very first one?
Yeah. Yeah, I mean, how do you get that?
Okay.
I
I will try.
Just tell us what are the inputs to get that thing.
Bayes theorem. The only thing. So let's see. Is the probability x given c given b. So usually, not to get it wrong, I do probability I'm sorry, I cannot talk and do it at the same time.
Yeah, okay, but based
No, but let me continue. Otherwise, I cannot actually. Okay, so now I can write it like 1 plus. Let's see. Well, I might be started to do wrong now, but let me just go quickly. What? So this is A, and then I have this term here. This is a social term, and then I assume, for the sake of the talk and the sake of having some mathematics, that the behaviors of the animals that have gone in decisions before me are independent. So my decision is not independent of them, but their decisions are independent. So I express this as a product of probabilities for single behaviors, and that in the end can be expressed as S because all the behaviors are the same. You have the behavior that corresponds to nx and the behavior that goes to ny, and you can write this down as nx. So you have a social part and a non-social part. So what happens is that when I apply Bayes' theorem, I call all this A. I do that trick, A. That's why in the end I say, oh, it's something I could manipulate, because I can manipulate things to manipulate these probabilities. And then I have a social term that I have some approximations to be able to mathematically express in a simple way. As I told you before, I can do things numerically that does not need to make these assumptions that give functions that are very similar. But basically, is to say there are two terms. Bayes' theorem is going to tell you that they come in a multiplication, and the reason why you get simple expressions is because I am assuming something about independency of animals before me.
Okay, it's a little bit more clear for me now. Still have reservations.
But, well, yeah. So it's the Bayes' theorem. You can use it to separate the social and non-social part, and you have to work your way.
Let's go back. You say that this is like fairly. I think before Google, all the
Well,
to search the web, use keywords. Look for keywords. The more keywords, the more. Then came Google and use the information. Who is the website pointing to that website? It's information of the others who is using. And then this changed completely. This improved a lot the learning process of the search. It's more or less the same. It's not looking at the thing, but at how the others look at the thing.
But my reasoning, you can call it.
I might be very fundamentalist, but for me, still this is not theory.
Wow. Okay.
This does not include
You are
negatives.
fundamentalist.
Yes.
So the difference between the difference between previous ways of creating some mathematics that relate to data was basically to say, okay, I will use this function, okay, blah 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 blah. Another data said, oh, I'm going to use this function, okay. The difference here, call it theory or call it not, is you start from a hypothesis. You make some mathematics that go with the hypothesis. You put it in a nicer way on a slide, and then you compare with data. And most of the time in my life are wrong. And I present on the slides the ones that seem to be working better. Okay. If it's not theory, I don't know. Hypothesis, mathematics, formulas, compare with data.
Theory and models mean very different things to very different people.
But it's not simple fits.
It's not simple fit, it's very elaborated fit.
It supports a hypothesis. Whenever, if the data does not correspond with this, I would say that the data does not support the notion that animals are using the behaviors of other animals to take decisions. So it's testing a hypothesis. Maybe it's not a theory. It's testing a hypothesis more than fitting data. In the end, it turns out actually that fits data very well, but it's more testing a hypothesis.
Okay. Any further question or comment? If not, thanks very much for that.